This computer.
Uh,
I'll just make sure. Do people see the red recording thing?
yeah, I think so. Well, I see it. I see it.
Wonderful. Okay, hello, year ones. Welcome. We hope you had a good week. Congratulations on getting in. My name is clap for them, please. You've done the hardest bit, which is good. Now you've just got to survive. And then in 5 years, you'll be the best. So what more could you want? Katya is here. So what we'll do is we'll go to the next slide. We know how competitive some of you are, so we thought we'd start with the quiz. So just chuck in the chat what you think the best medical show is.
I don't think you're actually sharing the screen, David.
Oh, I am.
You are?
I am.
Oh,
We got A, we
okay,
got
no,
B,
I'm stupid. Never mind.
We got E. We got A. We got E.
Great.
Come on.
Let's go. Good doctor.
Actually, good dog is pretty good as well.
We got an S. We got a smart fella. We got lots of B's. Okay. Okay, I'll tell you when. Anyone else? Okay, next slide.
Right. This is a very biased answer from, I think.
Sorry, everyone's wrong. Unfortunately, Scrubs is the best. It might be before your time. Then it goes House. I haven't seen Good Doctor, so I don't know. Grey's is definitely last. Apologies. You guys
House is pretty good.
But yeah, Scrubs is the best. So sorry if you got it wrong. Next slide. So today what we'll be doing is we understand that you might have been to a few of these. So we're going to try and keep it a bit more high yield than usual, but we'll start with some intros, both introducing you to the course, how everything works, some of our advice, who we are. We'll give you some study tips. We'll do some of the slides, and then at the end, or you can ask throughout if you have any questions about anything medical. I had absolutely no idea what was happening until about the 5th week, so please feel free to ask anything. Yeah, next one. Do people want to introduce themselves?
I'll start off. Yeah. Hi, nice to meet you guys. I'm Peter, and I live in Melbourne. I'm from Melbourne. Yeah, I am George Pig in the Pepper family. Yeah. There was a Mama Pig and George Pig left, so I chose George. I like most sports, especially basketball, and I'm pretty sad right now because Spotify got rid of some of the K-pop music. But that's about it from me. Nice to see you all as well.
I might go next then. So I'm actually from South Australia. I don't know if any of you are interstate, but I am Danny Dog in the family because I like dogs. I'm currently watching Friends. I listen to basically anything in music, but I like Nujabes, but you guys probably haven't heard of him. I also play a bit of badminton and games. And yeah, that's me. Okay, I'll pass it on to David, I think.
Hello, I'm David. I was born in Johannesburg, South Africa. Then I was in rural Queensland, and then I moved to Melbourne. I'm Rebecca Rabbit because I think she is a good symbol for Rebecca's everywhere and does good justice to the name. I'm currently watching The Office, which is fantastic. And yeah, it's about me. I'll give it to Katya. Welcome, Katya. You are muted. Do I need it?
Can't you unmute yourself?
What is happening? Not working? There we go. There you go.
You're muted.
Oh, Katya,
I think her mic works, actually. This is very sad. Works. Okay, well, maybe we'll just move on to Jono then.
Okay then. Yeah, sorry, Katya. Yeah, I'm from Melbourne, Daddy Pig, because I like dad jokes. And I really love food. I'm a big foodie. Favorite cuisines, top one's probably Vietnamese, followed by Japanese, close second, and Italian. But I do really like Korean barbecue as well. And yeah, no, I like all activities outdoors, so hiking, cycling. And it's been so good to get outside, like restrictions lifted. But yeah, no, lovely to meet you guys all. Thanks for coming.
I'll be Katya. This is what Katya sounds like if you don't know. Hi guys, I'm Katya. I'm from Melbourne and I am Miss Rabbit because I really like rabbits, even though I have lots of chickens. And Katya is killer at volleyball, which is true. And she's a big shot, so watch out. Okay, next slide. I mean, I'm a big shot. Watch out. So this is generally what our PSP will function as for you guys. So essentially, we're giving you slides as supplementary material and as resources for you to use. So a lot of our slides are made by looking on the internet, looking at textbooks and basically synthesizing everything. So it's a good place to kind of have it's a clutch, essentially. We'll be giving you some quizzes. Leon, our one member, he specializes in quizzes, so you'll be getting lots of those. Shortly, I'm going to make a Q&A sheet where if you have any questions, you can just pop it up there and we'll edit that. These recordings go on YouTube. You're encouraged to interact with us during the sessions, but your first week, so I assume you're very shy, but you mustn't be shy. Don't be scared. And essentially what we're going to do is we're also going to show you how to use different resources and techniques so you can study by yourself as well. So I know Andrew is going to do something about Anki for you, which is a kind of a memorization, active recall. If you watch YouTube, who's the dude on YouTube who talks about it all the time? Anyway, I'm sure someone knows, but yeah. And yeah, that's essentially all. So next slide. Essentially, this is what you're going to probably have got an Excel sheet and you probably don't know what any of it means. So you're going to have lectures, which is just when someone talks at you and they teach you content. You'll have tutorials, which are essentially your classes that were similar from school. You'll have a tutor and you'll go through classes and it's collaborative and lots of tutors are nice, so it's good. If you don't have a nice tutor, good luck to you. You also have workshops. Is it Ankin? Because if so, very cool. Yes, it is. Workshops which are done in group A or B, and that's mainly, it's a big overview of concepts. Sometimes you'll have Moodle quizzes. Sometimes you go into breakout rooms and do questions. It's quite broad. They had to really adapt that for Zoom, I think. And you have ICLs, which are really fun and really chilled, where they give you a case and essentially you learn how to be a doctor. So you go through clinical reasoning and how everything works. First sem is a bit strange because they kind of show you all the fields around medicine, but ICLs are quite medicine. I'll make you feel like a doctor. Next slide. If you're into textbooks, try not to buy any. There's lots of PDFs on Monash. Most people don't use textbooks. I like textbooks. These are some that I find helpful. Guyton and Hall is highly recommend. That's always going to be useful. Leningers is good for first sem. Weeders is not as useful. You might use that once or twice. Pathology, not as useful for first sem, but I used it a bit. And yeah, the lecturers in their lecture will tell you what textbooks they recommend, but it's really not necessary if you're not into it.
Yeah, I think generally like textbooks go into way too much detail. You'll be here all day if you try to learn everything. And like the lectures don't even go into that much detail, but it's useful every now and then just for some reference. So there's a bunch of textbooks on that drive. And I personally would recommend like the anatomy textbooks in Sem 2 when you have to learn a lot of anatomy. Also, there's a Netter's Atlas, which kind of has like all the pictures of like muscles and bones and stuff. That's pretty useful as well. And you'll find that on the PDF drive as well, I think.
Yeah. If anything, they're good pictures to look at. Next slide. How to study. Use PSPs. You can go to them. You can look at the slides. You can look at the YouTube recordings. Lots of people use Anki. I don't use Anki, but Andrew will talk to you about Anki at some point. Lecture notes should be your first starting point because most of your exam is going to come from lecture notes. YouTube is really good for conceptual understandings. Those are some channels. Osmosis is probably the best one there, but they're all very good. Googling is great. Try to be reputable sites. I always look at the authors and make sure they're doctors, and then I feel safer, but that's probably not great. But you want to you'll have an idea as you go along what's cool and what's not.
Yeah, I think a lot of studying is like sort of just experience. You'll get a better idea of how you personally study as you go. So Personally, what I do is like I sort of watch a bit of lectures and sort of see what they want you to know. But I find that lecturers often go into a lot of detail a lot of the time, and a decent chunk of them don't like get mentioned much again, and not that important on the test later on. So I think, well, what I do personally is like I note down what I think is the most important stuff, and that's just sort of common sense and experience, which isn't that helpful, I know, but like it's quite hard to tell sometimes. But what you can do is then you can check with your PSPs. So, you know, they're run by students, so they're generally quite high yield because, you know, we're lazy. We just want to do the least amount of work, and we took the exams and tests. So even if like some people don't even watch lectures, I know some people that don't do that at all. They just follow the PSPs, and they usually give you more than you need anyways.
Yeah, and a quick way if you want to do lectures is I'll give you lecture slides, make a OneNote, print the lecture slides in OneNote, and then you can key search words if you need. It's a good like way to keep notes, but not keep notes at the same time. Also, lots of people recommend making, if you do watch the lectures, which we'd probably recommend, lots of people recommend making summaries, because at the end you'll have lots of lectures and you won't really remember what's important in which one. So afterwards, it can be helpful just to 2 or 3 sentences, what's the main ideas. Yeah. These are your subjects. First, so you have anatomy, which is not really anatomy in Sem 1, but it's still very important. So you're going to go into more cells, sort of tissues, like what's epithelium, nerves, muscles, what do they look like, how do you classify them, sort of how do they work. You'll go into physiology, morphology, what they look like. Yeah, it's fine. Biochemistry is not fine. You have, you'll do a metabolism, which there's a lot of enzyme names you have to learn, and it's a bit not medicine, but it's okay. If you can have, they say that you can just have a general understanding of it and be fine. That's not true, but if that makes you happy, you can think like that. We'll tell you what's important and what you need to know. You also do central dogma, which is DNA, and then cancer, which is pretty cool. HEP is mindfulness and lifestyle tips, which you haven't met Craig Hassard yet, but are you in for a treat? So that's fun. That's more about you as well. It's like, how do you not burn out? How do you handle stress? HKS is social determinants of health. It's a societal perspective. It's cool as well. It's medicine in context, how social determinants affect health and society, and enjoyable. Med law, this is mainly big because there's an assignment on it, and it's like a cool assignment. It's about how do you apply ethics, what is consent, how do you do the least harm, weigh up the most good. So that's pretty chill. There's only 2 tutes on that, I think. Micro is you'll do types of infections and protection. So you'll feel a bit more like a doctor here than metabolism. And you'll go through different types of viruses, bacteria, fungi, parasites, resistance. You have to learn a couple of names. You'll learn the pathogenesis, so how the bacteria or the pathogen causes disease, and understand that. Mom is psychology. Pathology, you can read through this in your own time if you're interested, but pathimmunology is how injuries work. That's cool. Pharmacology, you'll learn the name of drugs and how they work. Physiology is physics and biology, so that's pretty cool as well. It's easier as well. Physiology is 2 things. There's like very easy acids, base, fluid stuff, and then you have the harder, cooler neurons, spinal tract stuff. Yeah, next. For your interest, the bottom is a breakdown of our exam for Sem 1, so you can see what's important. Biochem is quite important for some reason, but yeah, this gives you context and perspective, so you're not stressing about mom in your SWOT fact the whole time. You'll have assignments. The assignments are aggregated. You will either pass or fail them. Then you'll have clinical skills and written papers. Clinical skills are a bit weirder on Zoom. I think faculties themselves are trying to figure it out. Written papers, Sem 1 was just pass fail, but they did give you a scored mark, but it doesn't really matter. Sem 2 is a graded mark, but your WAM, so I don't know if you know how it works. First 2 years, you get a WAM, not really that important. It's important if you want to apply for scholarships or do some research stuff. So it's quite particular.
Yeah, WAM is basically GPA.
Yeah. So it's not going to affect you in the long term. In years 3 to 5, you get something called a Z score, which is basically an ATAR, and it ranks you. And then that's used to determine where you do internships, which you don't have to worry about now. And it doesn't really, from what I've heard, it doesn't really matter where you do. I stressed irrationally about it. It doesn't matter that much. New South Wales, they just do it randomly. So they might not do it when we get there. Does anyone have any questions about anything that's come up during the week, during the lectures? Anything med related at all?
Live questions.
You can unmute if you like. If you're too scared, you can write it in the chat. Or
you can always direct message us as well.
All our Facebook accounts are names. I'll give it 10 seconds, like 5 seconds.
How's the start to
the week has
been for you guys? Oh, sorry, you go.
Sorry. It's really uneventful, so I don't think anyone's gonna have any questions. We've done like one tutorial. That's it.
You got to get into more trouble, so it's more eventful. You got to break some things. Have you been into?
Trying my best. I really have been trying my best.
I'm very proud of you.
Thank
have you guys
so
been
much.
in campus? Have we
no,
been to
not we might for study, but we don't have any on-campus stuff yet. It's all online.
Okay, well, our first real thing was like yesterday. Campus is good. Can't complain. The clinical skill stuff, like you, especially, you're quite isolated now, so it feels like VCE, and I don't think you've gotten out of that. So some of you would have done IB. I don't think you would have gone. You don't really get out of that mindset until much later, unless you're in university, and you realize everyone's friends and there's no competition, and it's all good. So it's much more fun to study with people, and you actually learn more. So recommend that if you can, but otherwise, just do what you can. Hopefully, we'll all get vaccinated, unless you're anti-vaxxed up, which Katja will beat you up if you ask. So Katja like is doing something really. She'll tell you about it someday, but she's involved with the vaccination stuff. She'll tell you about it. It's really cool. Okay, we'll keep going then. Okay, so now we're doing some lecture slides. We'll try to keep it as brief as possible because we're aware that you've probably been to a few of these. And if you have any deviation, any other questions. Also, they're quizzes. I recommend that you private message Peter for the answers, just so everyone has a chance to think, and then you don't have to get embarrassed if you get it wrong. So that takes that away. Yeah, so Peter, I'll hand it off to you.
All right. Okay, so as David said, you guys probably saw a bunch of these, but I'll try to kind of skip through a bit of it. And I will be uploading the slides so you can kind of go back to it, but I feel like you could just skip some of it. So yeah, first two lectures you'll get, I believe, is cells 1 and 2. And I think in the lecture they describe like the modern cell theory and stuff. I believe it's not that important, but it's basically just memorization. So you guys can have a look at that if you want. This is the differences between prokaryotes and eukaryotes. Really the main things are like that prokaryotes DNA is circular, eukaryotes DNA is linear, and it's found in the nucleus, whereas prokaryotes have it free floating. And no membrane-bound organelles in prokaryotes, whereas eukaryotes have a membrane-bound organelles and a nucleus, and that eukaryotes are generally larger. So these are the 4 features that the lecture slides last year went through. I believe will be the same this year. And we'll go through them. So cell/plasma membrane. If you did Bio 3-4 in BCE, you would have covered all of this. I believe in Bio bridging course as well, you would have done this. So I'm hoping you guys know some of it. So it's a phospholipid bilayer. So it's made out of phosphate first page group, a head-to-head glycerol, and 2 fatty acid chains. Yeah. So on this right, this is how they orientate. And because the tails are hydrophobic, it's inside, so the water doesn't kind of attach to it, whereas the hydrophilic heads just touch the water. So the water will be like extracellular here, basically. And technically, water is technically in the intracellular as well, so there's water here as well. But basically, within that tail, between them, there's no water there. And this arises one of the characteristics of plasma membranes, which is that they are selectively permeable, right? Only small non-polar, because polar means hydrophilic, non-polar means hydrophobic. Well, we can kind of interchange those terms. So non-polar, well, it has to be non-polar because it needs to pass through the tails. That's why only non-polar and small molecules can pass through, simply diffuse, so they don't need any help. And polar molecules, they normally require, or they will require, facilitated diffusion. So they need a protein to kind of make way between the phosphate bilayer to get through either outside or inside the cell. And also there's this term called fluid mosaic model, which is just describing the plasma membrane. And that is, well, fluid means that the molecules in the plasma membrane move laterally around a single phosphate layer. Well, this is what I learned in Bio 3-4 and not in Med, so I'm not quite sure if they think differently in Med, but I believe it's the same. And mosaic is the fact that there's a lot of proteins, as you can see on the top right here. These are all proteins and glycoproteins, which are just protein with a carbohydrate attached to it. So you can see all these patterns kind of, and that's why we describe the cell membrane as mosaic as well. Yep. And then there's the nucleus. So this stores the genetic information, and a cell can have 0, 1, 2, 3, or more. So 0 would, like cells with 0 would be like red blood cells. They don't have nucleus. Cells like muscle cells, they need a lot of power basically, and the more nucleus, the better basically, so they have more. And within the nucleus, there's also some other things that you need to know. The nuclear membrane, nuclear envelope. This is like what encloses this thing called the nucleoplasm, which is just, as you can see here, like the plasm of the nucleus. And it's a double membrane with pores, so it's got pores, so you can kind of get out of it. And the chromatin, which is the DNA wrapped around the histones. So this is just DNA. And this is stored within the nucleoplasm, so like these things right here. Nucleolus, this is a site of ribosome synthesis. And there's a lamina and matrix. That's just definitions. I don't think you need to know too much into that. So, yeah. Okay, now there's the organelles. I've provided a really brief table here, right? And I believe that this is really the surface level you need to know. You probably need to know a bit more, but this is, if you're going to go into an exam and this is all you knew, you probably pass the test, right? But you probably would need to know a bit more, such as where are these synthesized, for example, or why are the differences between smooth and rough endoplasmic reticulum. But yeah, ribosomes, they make proteins, right? Mitochondria and mitochondria, energy production, ATP, smooth ER, endoplasmic reticulum, they have lipid production, and they also detox. And they have no ribosomes, which is why they are called smooth, because they look smooth, there's no ribosomes attached to it, whereas rough ER does, that's why it looks rough, quote unquote. And rough ER also makes proteins. Golgi apparatus, which is very important. This is where protein modification and export occurs. And layers, not layers, but different structures are also there. Lysosomes and security vesicles. Lysosome is also like, it's like a vesicle, but it's like, it has protein destruction. So when they engulf with it, they have like certain certain things inside it that basically destroy whatever they have inside. And this is made in the rough ER and Golgi apparatus. And security vesicles is help with extracytosis. All right, now this part, cytoskeleton, this is kind of the main part about in cells, because it's something that is very interesting, but also it kind of interrelates, it kind of like links with everything else that we'll be talking about in anatomy or in Sem 1. So cytoskeleton, this is made of 3 things: microtubules, microfilaments, and intermediate filaments. Microfilaments are also interchanged with the word actin microfilaments and actin myosin filaments. So they're just the same thing. Well, they're referring to the same thing, basically. So that's one thing you need to know, because they will be using these terms differently, especially in lectures. So you need to know that. And this is really important because the cytoskeleton basically moves the cells and the organelles within it, and maintains the shape, and also moves part of it, and also has specialized functions, such as being a spindle in mitosis and meiosis, which we'll be covering a bit later. This is a really good table that I basically copied off from some lectures textbooks and some PSPs. As you can see here, the microtubules are the biggest. And intermediate filaments are middle, and microfilaments are the smallest. If you want to help memorize the size, I guess like tube, it felt like that seemed the biggest, intermediate, medium, so that's middle, and filaments smallest. You might need to know this, like the size, just because you might get asked, like maybe like why, like other size is different, but we don't need to memorize it as such. And the more important thing here is that the microtubules are made out of this thing called tubulin dimer, and microfilaments are basically made out of actin, which is why we also call them actin filaments. And intermediate filaments, they are made out of several different proteins. So for example, it could be made out of keratins, but there's other things that it could be made out of. So this kind of depends on where you are in the body. So microtubules, the functions of that is to give the cell the shape, but also control the substance transportation in and out of the cell. So as you can see here, this is a simplified diagram of the dimers that we were talking about before, dimers because I think it was two, of alpha tubulin and beta tubulin. And in mitosis, they basically make the centrioles and make the mitotic spindle. And in cell motility, they make up the cilia and flagella. And going into cilia and flagella, flagella are like hair structures extending from the cell surface to move the whole cell. And they are longer than cilia normally, whereas cilia are smaller, and there's two of them, motile and non-motile. So motile aids in the movement, so it's kind of the same function as flagella, but just shorter. Whereas non-motile is just like sensory organelles, so like in most other cells. So really in trachea and fallopian tubes, I believe is the only examples that we were given about cilia. So I guess that's all you need to know about. And yeah, the more important thing is that cilia and flagella kind of do the same thing if you're talking about motile cilia, but they are shorter. And they also both have a 9+2 structure. So if you look on the right here, this is the structure of basically the microtubule. So both cilia and flagella. There's two here, and then there is 1, 2, 3, 4, 5, 6, 7, 8, 9, if I can count right. Yes, 9. So 9+2 is the structure that all microtubules will have, right? And when you have these 9 microtubule triplets linked together, we get centrioles, which is also a very important thing in anatomy. So right here is a very good picture of how they orientate. So that is a centriole. As you can see, there's triplets, and there'll be 9 triplets of the microtubule going around, forming the centriole. And then this will be aligned like 90 degrees from each other. Like this creates a 90-degree pattern right here. And this is used to help, well, this is used to help cell division. And that orientation right there is basically what we call a spindle formation. They are found in pairs and orientate 90 degrees. So centriole is very important in a cell cycle, which we'll also go into a bit later. Now, let me just check. Okay. Good. So now we're going to microfilaments. Right. These names, like they're kind of confusing when you first hear it, but you'll get used to it. Microfilaments, as we said, actin filaments are what they're made out of, but also you can also say actin and myosin, sorry. Again, they just mean the same thing. And the thing that separates microfilaments from others, if you could remember, microfilaments are the smallest. This also kind of helps them assemble quickly, but also disband quickly. So they're kind of good to just kind of like, if you need it, you get it, and then they just disperse. Good for motility. So actin is abundant in basically all your cells, whereas myosin is scarce except in skeletal muscles. And actin are also globular, and they're in double helix, as we can see here, kind of like how DNA kind of works, double helix. And they are a high concentration in the cell periphery, which you'll be able to see later when you talk about the muscle cells or tissues. And again, as we said, important for cell motility. And myosin, because they're so scarce, they actually can't be seen under a microscope normally, unless it's under, or unless you're looking at our muscle cell, because as you can, we'll be talking about later, but there's a lot of them in skeletal muscle. So microfilaments, they have 5 main arrangements. First is just being in skeletal muscle. One is beneath the plasma membrane, called actin. So it's like, so this is when it helps with cell movement and endocytosis, exocytosis, so like transportation of good substances going in and out. The third one is movement of cytoplasmic organelles and vesicles and granules. Cleavage of mitotic cells, so this is mitosis, part of mitosis. So this forms a ring of filaments and it pinches into the cell into 2, which we'll be talking about. And it's also, it could just be scattered in the cytoplasm. So those are the 5 main arrangements. I believe there are only 5. And you can kind of see how different arrangements also have different functions. So now going on to intermediate filaments. These are more permanent. So compared to actin, myosin filaments or microfilaments, they are more permanent. So these are better for maintaining shape, because if it's good for motility, they're disbanding quickly, right? But if you want to maintain shape, you need to be permanent. That's what intermediate filaments are. And as we said, there's different types depending on the protein subtype, basically. So there's keratins, that's what we talked about before. There's also bimetin, desmin, neurofilaments, blah, blah, blah. This was off the lecture, so you can have a read through it. But yeah, that's just, I think you got, the main thing is that there are different varieties depending on which protein subunit you're talking about for intermediate filaments. So first question is pretty simple. Where does protein synthesis occur? I'll give you guys roughly 20 seconds. Looking good. All right. Yes, the answer is A. Looks like you guys paid attention in bio, bridging course of bio 3-4. But yeah, it's ribosomes. Ribosomes are where protein are produced. They could be free floating or attached to raffinol. And here is a good diagram of explaining how that works. And nucleolus is the site of ribosome synthesis. So this is where ribosomes are. And Golgi apparatus is where proteins are modified. So after proteins are made at ribosomes, this is where it goes to, and it gets exported through there. All right, second question is, what structure does cilia have? Confident, I guess. That's good. Yeah, another easy question. C, 9 + 2. Just remember this diagram. This diagram helped me a little bit, just memorizing a little bit. Like, it's pretty obvious, but sometimes you might just forget it. But if you just know it once, it's something that you want to keep for a while. So that's the basics of cells. Now we'll be talking more about the cell cycle. So the thing we just talked about before, I personally believe that it's not as important. Like, it's things that will help you with the basis, but it's not really the most important thing in anatomy, Sem 1. This cell cycle and proliferation and death, this is more the content heavy part. So cell cycle, what is it? Well, cell proliferation is just making new cells. In growth is cell production, and replacement, so cells do replacement. And that's when cells which are worn out or damaged are replaced. And this can be kind of done in different ways, but there's two main cell cycles. There's two cell cycles that we learn. One is mitosis, second one is meiosis. And on this right here, this is basically how all cell cycles look like, except the mitosis part, depending on if you're doing mitosis or meiosis. And we'll explain it here. So there's interphase, which is G1, Z0 if you need to go there, S, and G2. Those are the phases. And there's the mitosis phase, and then there's cytokinesis. I believe that last year we were told that cytokinesis technically isn't part of mitosis, but it's kind of like, I don't know, like some studies say they are, but some studies say they don't. I think we learn it as separate. I don't think it matters too much. So cell cycles in any organism is basically the same, except that the timing varies. So bacteria, it could be as little as 20 minutes, whereas for us, it could be more than 24 hours. But kind of the fundamentals stay the same. So going into interphase. So G1 phase means gap 1 phase, and G0 means gap 0, and then S means synthesis, and G2 means gap 2. So G1, this is the longest period within the cell cycle. And if the cell is functioning normally, correctly, they want to go to S phase. Right? If the cell was in G1 and they're completely healthy, then they want to go to S phase normally. Cell growth and organosynthesis occurs as well, except DNA, I believe that is, sorry, that presentation is cut out. And within G1, it basically preps for the next phase, which is S phase. Now, there is a case where you could not be going into S phase, and that is when you do end up going to G0 phase. So G0 phase, again, sorry, to cut out. This is when cells cannot divide, for example, nerve cells. And they come to G0 phase, which is just, they just don't do the cell cycle. And they never leave the G1 stage. So they're called and termed G0 stage because they never enter the X phase. They're always in G1 phase. Next one is now the S phase. So as the name suggests, it's when DNA replication occurs. So before here, at G1 phase, cell is still growing and like organelles are being synthesized, but DNA isn't being doubled or being synthesized anymore. Whereas in S phase,
sorry, keep going.
Yeah. Whereas in the S phase, that's when DNA replication occurs. So, yeah, did you want to say something, David?
I just want to ask a question. Does anyone know what sort of cells are in the G0 phase most of the time?
Isn't it nerve cells or something like that?
Why? And what does that? What is the implication of that? Do you, sorry,
I have no idea.
If a cell doesn't, if a cell cycle is arrested, what does that mean? If it doesn't go through the cell cycle, what's the point?
If it's damaged, we can't replace it.
Essentially, yeah. So if you damage nerve cells, it's genuinely permanent. What other cell that you damage do you reckon is going to be pretty permanent? Can you think? This is extension. So cardiomyocytes, which are your heart muscle cells. So if you have a heart attack, it's why people usually don't recover very well. So there is actually, they did find there are a few stem cells there, and some of them do go through this, but it's quite limited. And that's why most people, when they have heart attacks, their heart's fibrous, which means there's strong fiber that doesn't allow it to expand, and it never really heals properly. So that's just some reason why some of this is important.
Yeah.
Sorry.
No, that was good. Yeah, so that was that. Now, S phase. So as we said that we have the doubling of DNA basically, and during the S phase, the result of the S phase, you should get 2 identical chromosomes of each chromosome type because you're doubling the DNA, right? And we have this point called the centromere, which is the point where 2 of the sister chromatids are held together. So that's an important word you should know. And now we finally enter G2 phase. So after S phase, we go to G2 phase. And here is when the cell prepares for mitosis in this case, because we're referring to mitosis, but it could be meiosis. And during G2 phase, it double checks for any DNA replication errors because DNA replication errors is detrimental. If we stuff one up, we need to kind of fix it. And also at G2 phase, further growth occurs. So like they're trying to make sure that the whole cell is basically double the content within itself. And also microtubule, which we were talking about before, the spindles also start to form in G2 phase. So they're getting ready also to kind of get into mitosis. And what the hell is mitosis? Well, it's basically to produce 2 genetically identical daughter cells from one mother cell. That should be saying mother cell there. Yeah, so as we talked about before, there's like this doubling that's occurring in the interface. That's all to basically try and get two genetically identical daughter cells if we are entering the mitosis. So in mitosis, there is 4 stages technically, because if you count cytokinesis as a separate one, there'll be 4 stages. And that is prophase. Technically, there's early and late prophase, metaphase, anaphase, and telophase. So I don't know if I heard this from, but I heard this from someone. It's like PM, so like Prime Minister at Carnival, is a thing that's helped me memorize the steps and the order. But yeah, if you can come up with a better one, just do that. Yeah, and cytokinesis is basically when the cell actually divides. So technically, the steps of mitosis, by the end, you don't actually have 2 cells technically. You need to go through cytokinesis. That's when the cell actually divides into 2 separate ones.
Sorry, keep going.
Oh, yeah. Okay. Yeah, and prophase, there's 2, as we said, early and late. In early prophase, this is when chromatins of the DNA basically begins to condense to chromosomes. And during the late prophase, that's when nuclear envelope, which is something we talked about before, and the nuclei starts to disappear, which means that the chromatins or chromosomes now that were within the nucleus basically are kind of just spread out. They're not within that anymore because the nuclear envelope disappears. And yeah, the chromosomes are fully condensed, which means that spindle fibers start to also, and spindle fibers also start to form. And at this stage, the cells are 4N. So there is 2N and an N. Sorry, guys. Yeah. Yeah, so there's 2N, which is, we call it diploid. So that's just the normal amount of chromosomes, basically, a cell should have. Whereas N is haploid, so half the amount of chromosomes that you should have. So 4N means you have double the amount of chromosomes you should have. So this phase is basically preparing a cell to enter the next stages. That's why you can remember P for prepare. Next is metaphase. And this is when the chromosomes align on the metaphase or the equilateral plate. And this is like right here. It's basically the middle. And the spindles, which were growing before, they kind of grow even more and form attachment to the chromosomes, basically. And at this stage, the chromosomes are the most condensed state ever. So when we do like karyotypes, which is like basically you're looking at the DNA and the chromosomes, this is when it's done, because in normal times, you actually can't see the DNA really well because it's not condensed. But in this stage, it's the most condensed, so you'll be able to see it pretty well. So M for middle, that's the way I used to memorize it. It's coming to the middle, metaphase. Yeah, that's when the spindles attach to the chromosome. Yeah, so like this. Then next is anaphase. This is when the sister chromatids, which were held together at this by the centromere, are pulled apart by the spindles, basically. And they move towards the poles of the cell. So the poles of the cell, I think they talk about this one. It's the opposite ends. They get like pulled apart. So you can memorize A as apart, I guess. Then there's telophase. So after we basically separated the sister chromatins, the chromatids, we have to kind of now enclose them separately because we don't want to have double the amount of cell content in one cell. So the nuclear envelope starts to reassemble. So basically they take in the chromatids that they separated, and the cell membrane constricts at the middle. So it constricts right here, and then the chromatins start to decondense into chromatids. That should be saying chromatins. And you can memorize telophase T for 2 cells forming, I guess. And that's the end of mitosis technically, right? So it's still attached. You want to say something, David?
No, keep going. Sorry, I didn't mean to. I'll do it when you're finished.
Okay. Yeah. And then the cytokinesis, which is when the 2 cells are split for short. And that is mitosis. That's the end. So you will see that you have this there's two identical daughter cells from one mother cell, which has gone through interphase already. So she's got, well, the cell's got double the amount. And they go through this, split the double amount, then make two cells, and then they get two identical daughter cells.
When are the chromosomes most condensed? In what stage?
Did you want me to answer that one?
No, then. I know you know.
Metaphase?
Good job. Why is that important? What is the application of that? Anyone?
You can see it better. Like
That's
all the best in metaphase, and so that they can be pulled apart.
yeah, great. Why do you want to see chromosomes and pull them apart? What does that tell you? Can you think of a clinical application of that?
Maybe like checking for like genetic disorders or something like that, if there's errors in the chromosomes, or if
Right.
there's like an irregular number or something.
Great job, yeah. So often what people do is they karyotypes, wonderful. Did you do this in bio, or are you guys just super smart? I'm just going to assume you're super smart. So often what they'll do is they'll take some amniotic fluid out of the mother to check the chromosomal constitution of the baby, see if there'll be any genetic abnormalities, and through a karyotype. So good job.
Smart. Okay. So now we'll talk about meiosis. So mitosis is just making two daughter cells from one mother cell. Meiosis is basically producing gametes or sex cells. So only the germ cells, which produce the gametes and sex cells, only go through meiosis and mitosis. So normal cells don't go through meiosis because they don't need to make sex cells, basically. And the germ cells, I believe, are labeled immortal. That's because they kind of form links between generations. So that's a fun fact. And meiosis has two stages. So meiosis 1 and meiosis 2. And meiosis 1 is literally the same thing as mitosis, except in two stages, metaphase and anaphase. And meiosis 1 also, before meiosis 1, sorry, they also go through interphase as well, meaning that the cell already has doubled itself, basically, in content. Okay, I don't know why they have meiosis 2 there, but yeah, that's a wrong diagram. But meiosis 1, not meiosis 2. So as we said, meiosis 1 is basically the same except at metaphase and anaphase, okay. But that's like the big differences. There are small differences within each, I think, but I don't think you need to know that much detail. But in this one you do, which is in prophase 1, so meiosis 1, meaning it's prophase 1. Sorry, I'm confusing myself. Meiosis 1, so that's the first meiosis. So we label prophase also as 1 because it's part of meiosis 1. And during prophase 1, the homologous pairs are aligned, and this is when crossing over occurs. So in mitosis, there's no crossing over, and crossing over is basically swapping of generic material. All right. In mitosis, we don't need this. We don't want this at all because we don't want to create the same two identical daughter cells. But if you're crossing over, you won't occur, right? So in this case, you don't want it. So you don't want it in mitosis. But in meiosis, you do because you create sex cells and you want a new baby. You don't want a complete clone of yourself, right? Well, you can't technically either. But basically, this is when crossing over occurs. And that's a little bit of a difference there in prophase 1. In metaphase 1, it's again similar concept, except the homologous pairs line up on the equator and they line up either side. So not side by side like mitosis. So they line up like this instead of like this, if that makes sense. Yeah, it's better if you see a diagram, but it's, yeah, you can see that it's lining up at they're lining up either side, not side by side. And in anaphase, the homologous chromosome pairs are separated. So not the sister chromatids, but the pairs are separated. So before in mitosis, the sister chromatids were separated, but these are sort of attached, the chromosome pairs that are separated like this. And so instead of these individual ones getting separated, the pairs are just separated. So they kind of still technically 2 chromosomes. So that also means that the first order cells has 23 chromosomes. So that's the difference. That's the biggest difference right here between mitosis and meiosis, is that you don't have 2 identically similar or same. What am I saying? 2 identical daughter cells at the end, whereas in mitosis you do. Then telophase is the same. Cytokine is basically the same, except, as I said, at the end you have 2n, so haploid, so half the amount of chromosomes that you normally have in meiosis, whereas mitosis you have 2 identical daughter cells, so 2 times 2n, which is diploid, so the normal amount of cells, chromosomes. So this is because we did not separate the chromatids as we talked about, instead just the chromosome. So that still counts as a haploid number of cells, a haploid number of chromosomes. Then we go into meiosis 2. So it's the same thing as mitosis again, except it's the haploid number of cells, haploid number of chromosomes. We have 2 daughter cells in meiosis 1, right? As we talked about before, the end of the 2 right here. So now that those 2 enter meiosis 2, basically separately, right? And at the end, you get 4 haploid daughter cells, right? So they do not have 23 chromosomes, they have either 46 chromosomes, they only have 23 chromosomes. And there is no interface between meiosis 1 and meiosis 2. So you go straight into meiosis 2 from meiosis 1. This is the difference. This is a really good table that I think someone found. Yeah, so at the start, mitosis starts off with 2n, same as meiosis. But then at the end, mitosis has 2 diploids, so the same number, the normal amount of chromosomes, 46. Identical daughter cells, whereas meiosis has 4 haploids, so, and they're also non-identical daughter cells. That's the biggest difference. And the chromosomes, they are obviously the same in mitosis because they're identical daughter cells, whereas meiosis, there's only half of the number of the chromosomes, so they can't be the same. The purpose also for mitosis is more for growth and replacement, whereas meiosis, we talked about, is the production of sex cells for sexual reproduction. And the stages are 1 cycle of the PMAT, and meiosis has 2 of them, but there is no interface between 1 and 2. Oogenous and spermatogenous, we covered this in one of the lectures, but I think this is kind of the thing you need to know more. So it's basically, oogenous is when eggs are made, spermatogenous genesis is when sperms are made. The similarities are that they're both meiosis because they're both sex cells, and they also have extensive morphological differentiation, and also cannot survive for long if fertilization does not occur. But the differences are, there's 2 differences. One is the mitotic differences. So oogenesis are asymmetrical. So only 1 egg is formed from 3 polar bodies. Look here, if you can see here, the result of it is 1 egg and 3 polar bodies. So we call that asymmetrical, but it's not symmetrical. Whereas sperm, basically, it's the same. You get same 4 spermatids. So after you go through meiosis, you get exactly the same, you get 4 of the sperm. And the other difference is the maturation difference. So it's interrupted at 1 or more stages for eggs, whereas sperms are rapid and uninterrupted. So males, they go from puberty to death. They just keep producing it, whereas females, they're from prenatal until puberty, as in one of the stages, and you don't make them after some age as well. Now, that's important. Meiosis and mitosis, especially meiosis, is important because that helps with genetic variation. So we talked about crossing over during prophase 1. This allows for genetic variation within us. Right? If we're getting the same outcome every single time, we're going to get the same amount, the same people basically, theoretically. But crossing over kind of helps mix up the genes, therefore we get genetic variation. So during prophase 1, you get recombination, so shuffling of alleles, during meiosis. And then there's this other thing called independent assortment, which is when we were having the homologous chromosomes randomly line up. When they're lining up, they're randomly lining up. So there could be up to 8 million possibilities. That's also genetic variation. Random fertilization, as we talked about, there's different sperm and ovum in different people, and there's up to 8 million possibilities when we do sexual reproduction. So those are 3 ways that genetic variation occurs. And obviously, it's not all good in life. So there's non-disjunction events, and these are really bad. I believe this is also talked about in biochem and in the later lectures. Basically, non-disjunction events are when the chromosomes do not separate like they're meant to be during normal meiosis. So this could be one where it occurs in meiosis 1. So it did not, for some weird reason, did not divide like it's meant to. Then now you're going to have basically these 2 cells that technically don't have any chromosomes, whereas this one has double the amount that you're meant to have. Whereas it could also occur in meiosis 2. So the first part was right, but in the second part, this guy didn't divide properly. Then you're going to have 1 cell, and then one cell that's empty basically with chromosomes, 1 cell with double the amount. That's just all bad. You just can't have that. You can, but it's really detrimental. So most non-disjunction events do lead to miscarriages. And as you can kind of see here in this picture, non-disjunction in meiosis 1 is worse than meiosis 2 because this empty cells kind of goes through another step and kind of produce more problems. Whereas in meiosis 2, it's kind of like you can kind of technically get 2 cells that are perfectly fine. So that's why it's more detrimental to have meiosis 1 non-disjunction. So as we said, there's miscarriages normally if non-disjunction occurs, but there are some events where you can have a baby that does survive for birth. And this is more covered in biochem, so I won't go to too much detail, but there's Down syndrome, Edwards syndrome, Patau syndrome, I can't pronounce this, Klinefelter syndrome, and Turner syndrome. Sorry for that, but yeah, that's it. And now we're going to be talking about the cell cycle checkpoints. And these exist in the cell cycle to make sure that if the cell division is not working correctly, it is corrected. And if it cannot be corrected, the cell will die. But if this does not occur, so if all these checkpoints do not occur, then you could possibly get serious problems like cancer. So I think someone mentioned in the chat before, but cyclins and cyclin-dependent kinesis, this is basically it. I won't go into too much detail. You guys could probably have a read over a bit later. But yeah, this is how it works. Now, I believe this is a bit more important than the CDKs because in our lecture, you spend more time on this. But there's 2 important inhibitors, proteins, that affect the cell cycle. First one is P53. And the name is because, okay, it should be 53 kilodaltons. It's just the name because of the size. And we call these negative regulators because it arrests the cell cycle if an error occurs. So P53 normally activates transcription of the inhibitors of the CDK cyclin kinesis, which will block the transition of the cell from G1 to G1 to S. So when the P53 is inactive, the cell enters the S phase. If it's active, it can't go through because it's an inhibitor. Then there's also RB, so retinoblastoma. It's because the name is because that's what causes, the defect in RB causes the disorder of retinoblastoma. And this inhibits the cell cycle progression from G1 to S phase as P53. So when RB is inhibited, the cell can enter the S phase. The same thing, because they're inhibitors, when they are inactive, the cell cycle continues. If it's active, then they stop the cell cycle. And there's also cell death, apoptosis, and necrosis. Apoptosis is apoptosis, I can't pronounce it. Programmed cell death. It's a normal process in most cells, and it allows for a balanced number of cells in tissue. So you might have too many, or you might have too little, right? If there's too many, apoptosis occurs. If there's too little, then cell growth occurs. And then there's necrosis, which is the bad one. So it's unprogrammed, unexpected cell death. So you didn't want the cell to die, but it died. So if it's normal, you have certain triggers like DNA damage. So if the cell is damaged, right, without trauma, so just damaged, then the cell will be like, oh, okay, kill this cell because it's useless. It's not worth anymore. And it could also be extrinsic, where it's like it's affected by toxins or infected or hormones or growth factors are telling this cell to die, then this will occur. So this is kind of pretty evident in when we talked about the shaping of the fetus in the hands. So apoptosis occurs so you can get rid of these cells to actually make the hand. Before it's just like a fist, but then you got to actually kill some of the cells to actually get the hand. So that's when a good example of it. So the process of this is, firstly, the cell shrinks, right? Then the chromatin condenses. Then the nuclear envelope dissolves and the DNA is fragmented, so that things inside are just getting destroyed, basically. And then after it's kind of all destroyed, the blebbing occurs, so the cell membrane buds off, so like the membrane kind of buds off like this. And these are called apoptotic bodies. And then these kind of bodies are phagocytosed by other cells. So they don't kind of just float around, the other cells come in and they're like, all right, we'll clean this. So they're kind of like making sure that the body is clean. Now, necrosis, this is bad because, as we said, it's unprogrammed cell. So before it's all programmed. So you're like, oh, good, we know what's going to happen, basically, right? It's death, but we know it's happening. So we can kind of make sure we do something about it. Whereas necrosis is not. It's kind of unprogrammed, so it's normally triggered by external factors, normally trauma, so injury, infection, cancer. And basically the cell swells up. Before, the cell was shrinking. Now the cell's swelling up, and then this bursts. So instead of kind of like being clean and getting into little bodies, so it's like rubbish bins for the rubbish guy to collect, this is just like bursting. It's kind of throwing rubbish on the floor, right? Then this could damage nearby cells and cause inflammation, which is not something you want. So a lot of times you would actually require surgery because the phagocytes, which are the things that actually, cleaned up the rubbish basically in apoptosis, cannot detect the necrotic tissue. And that's because there's less chemical signaling, which will mean that there's dead cells and dead tissues building up without being cleaned, which is really bad. So that's why necrosis often needs a surgery. This is a good simple diagram, table between apoptosis and necrosis. As we said, regulated, unregulated, unprogrammed, programmed. That's really the main differences, and how the necrosis could cause damage because it swells and then bursts. So some final questions. When P53 and RB are activated, it controls the cell by Yep, answer is A, inhibiting progression. So we said when the P53 and RB are active, they inhibit. So when they're inactive, they allow the cell cycle to occur. Or if it's something that you're meant to have been, something where the inhibitors are meant to be active but aren't, this will cause uncontrolled cell division, which you'll learn more about in biochem. Okay, which stage of the cell cycle does cell growth and organosynthesis, the doubling of the cell size, occur? G1 phase, G0 phase, S phase. Yeah, so G1 phase. So as we said, the doubling occurs in G1, whereas the DNA replicates in S phase. And G0 is just obviously not divided. And that's all for anatomy. There's a lot, but that's it. So thank you. Now we'll move it to John.
Can we just give Peter a clap for like speaking non-stop for like almost an hour? But yeah, no, once again, I just wanted to say like, yeah, seriously, congratulations to you guys. Like all the hard work from high school has paid off. And yeah, so glad you guys are in medicine able to join us. Yeah, don't worry about this year. You guys are gonna ace it. Yeah, so don't stress at all. Got any questions and stuff, feel free to ask us. So I'll be going over physiology today. Already going a bit long, so I'll make this nice and short and quick. So physiology, I think your lecture is cells and body fluids. It isn't scheduled until Thursday. So this may or may not be new to you guys. Yeah, no, what Andrew says is like, right, yeah, so much content in uni. Yeah, next slide, please. Yeah, so the first thing is fluid distribution. So H2O, so water in the body, actually has a concentration itself. So when it's just pure water, it's about 55.5 moles per liter. And so 60% of the body mass is water. 2/3 of this is intracellular, so meaning that the water is inside the cells, and then 1/3 of this is extracellular. And so extracellular fluid, you can find this in the plasma, so basically your blood, or interstitial fluid, which is sort of between cells and between the blood vessels. And your main intracellular ions and extracellular ions are there, but don't worry too much about them. It will be covered more once we get onto sort of like nerves and stuff. Next slide, please. Cheers. Yeah, so diffusion and osmosis. So these are two things that you'll learn. So basically, one's the movement of solute and one's the movement of solvent. So solute, it's basically the particles, what you're dissolving, right? So if you put salt into a cup of water, that's your solute. And the solvent is the liquid, so water in this case. So with diffusion, right, it's where dissolved particles or solute moves from high to low concentration. Osmosis is slightly different in that it's where the solvent, so water in this case, will try to move across membranes from low to high solute concentration. Next slide, please. Yeah, so diffusion is where particles move down their concentration gradient from area of high to low concentration. So if you sort of think about when you put some dye in the top of water, right? The dye isn't just going to stay in one place, it's going to start to spread out. And eventually, over time, it equilibrates. So diffusion increases with a few of those different factors, which are listed there. Yeah, but don't stress too much about that. Yeah. Next slide, please. Yeah, so I'll touch base briefly on membrane permeability. So membranes play a big part in cells in our body. So cell membranes, they're what we call semi-permeable membranes. So they allow some molecules to pass through whilst blocking others. So some examples, molecules such as urea, it can pass through easily. Then there's sodium and potassium. They're effectively impermeable, meaning that it does diffuse through, but it gets pumped back by some proteins, which we'll learn more about later on. And finally, there's molecules that are impermeable. Yep. And yeah, osmosis. So when you have a solution that's sort of separated by a semi-permeable membrane, right? And so in this case, the solute, so what is dissolved is the membrane is impermeable to that. So the solute can't pass through. So in this diagram, we have high solute on the right and low solute on the left. solvents, and basically those solid particles coming through. But the membrane is permeable to our solvent, which is water. So basically what's going to happen is that our solvent is going to move from area of low solid concentration to high concentration. So you can see it's going to move to the other side of the membrane. So here are a few terms. We have hypertonic, so it's when it's low in solid, hypertonic, high in solid, and finally isotonic when the solid is equal. So move from hypertonic to hypertonic. Right. Yeah, so some few core concepts and few terms that you'll learn for osmosis. So osmolarity, it's the number of solids per liter. So the unit is osmoles per liter. And the difference between osmolarity and concentration only measures the sort of number of one specific particle, but osmolarity doesn't discriminate. So you add up the concentrations of all your different particles. So for example, if you had NaCl dissolved in a solution, and let's say potassium as well. Well, for concentration, you figure out the number of molecules for one substance, but for osmolarity, we're going to add all of them up. So for red blood cell, the osmolarity is about 300 milliosmoles per liter. So there's about 300 millimoles of different particles in a liter. Osmolality, sort of similar, but slightly different, got mass instead. Finally, osmotic pressure is the pressure we have to apply to solution to stop the solvents coming through the membrane by osmosis. Next slide. Yeah, so effective osmoles. So these are solids that don't move across the membrane, right? So earlier on, we talked about the membrane being semi-permeable, so some particles can move through, others can't. And so these generate osmotic pressure. Then we also have ineffective osmoles, so these are just solids that can pass through the membrane. And something to note is the dissociation constant. So basically what this is, if AB dissociates into A, right, and 75% of it does, then the dissociation constant is going to be 0.75. So got a quick question here. So AB dissociates into A and B. And so the concentration of AB is 1 mole per liter. So if the dissociation constant is 0.25, then what is the osmolarity of the solution? So have a crack at this question, guys. Okay, I'll go to the answers now. Yeah, so basically 25% of our AB is going to dissociate, right? So that means 25% of AB is going to be A plus B, while 75% is going to remain as AB. So 25% is going to be split into 2 molecules. So we're going to get 0.5 osmoles from that. And finally, 75% is going to remain as 1 molecule. So we're going to multiply that one. So we end up with 1.25 osmoles overall. Does anyone have any questions about that concept? Okay, yeah, we'll continue then. So tonicity, this is a related concept to osmolarity, but slightly different. So tonicity only counts the number of non-diffusible or basically effective osmoles per liter. So remember, effective osmoles are those that causes water to move, the ones that cause the osmotic pressure, and the units are the same. So non-diffusibles, basically they're impermeable or effectively impermeable for membranes. And you'll note that isotonic does not always mean iso-osmolar. So we'll go into this in more depth. So hypertonic, isotonic, and hypertonic. Yeah, so here are some different solutions, and we'll sort of go through this. So pure water. So all of these solutions, so if we put a red blood cell, right, into a bath of water, red blood cells, they're permeable to pure water, right? So on the inside, red blood cells have an osmolarity of 300, whilst the pure water solution has 0, right? So, so like David said, this is quite high yield. So yeah, feel free to copy this into your notes. So with pure water, it's hypertonic to the red blood cell. So hyper meaning less, so less tonicity. It's got less effective osmosis. And so what's going to happen, right, is because, remember, with osmotic pressure, our solvent is going to travel to an area of high solute concentration. So water is actually going to migrate into a red blood cell. Peter, if you could go to the previous slide. Yeah, so you see for the first diagram, hypertonic. For the third diagram, which is hypertonic, water is going to migrate into our red blood cells based on osmotic pressure. And what that's going to do is it's going to cause the red blood cells to burst. So we don't really want that now. It's pretty disastrous for human beings. So if you go back to the next slide, you'll hear about 0.9% saline solutions. So these are all sort of different intravenous solutions that they can put you on in hospital. So 0.9% saline, basically it's just salt water. Has the same osmolarity as a red blood cell. So what happens when 0.9% saline is injected into our body, right? So those saline molecules are effectively impermeable to the membrane, so they can't cross over, so they're going to stay outside of the red blood cell. But basically, because we have the same number of particles inside and outside the cell, same concentration, so it's isotonic. So there's going to be no osmotic pressure, so no net movement of our solids. Yep, that's right. So effective osmoles are those particles which are impermeable to the membrane, so we can't pass through them. So could you go back to the previous slide, please? I'll just briefly touch on dextrose and glucose. This is quite important as well. So it's same as sodium chloride in that it's the same osmolarity, so 300 milliosmoles, and it's but the difference here is that glucose and dextrose, they get used up by cells in cellular respiration, right? So initially the osmolarity is equal inside and outside, but over time, as our glucose or dextrose gets used up, well, then the concentration is going to drop, right? So as the concentration of glucose reaches 0, then it sort of has the same effect as pure water. So eventually it does become hypertonic, so same as pure water. But in this case, glucose, it doesn't harm us. And the reason being is that this process is gradual and spread out across the whole body. So it doesn't happen immediately, so the cells have time to adjust. So what this does, it increases our extracellular fluids and intracellular fluids. Whereas the saline, because those salt molecules stayed outside of the cells, it only increased our extracellular fluids. And finally, we have urea. So urea, once again, same osmolarity initially, but the difference here is that urea is permeable to the cell membrane. So we'll go into more depth in this in the next slide.
Could I ask a question quickly?
Yeah, go for it.
People said they like house. In a house episode, someone had hemolysis of their blood, which means water went into the blood and the blood's like exploded. Does anyone remember how that presented for the patient? You ever think? Pretty tough. What's released when red blood cells explode? What's in red blood cells? Any guesses? So you have iron in your red blood cells. So if you have hemolysis, it means your pee is going to be brown. And that is in a house episode. So, fun facts, because of the iron.
Very nice. I need to watch that episode. Which one is it?
Watch Scrubs instead.
Yeah. So tonicity, we're going to look at an example here. So imagine we've got a bath of urea and we're putting a single red blood cell into it. So the osmolarity of our red blood cell is 300 milliosmoles. Same with our urea. So they are iso-osmolar. But the cell membrane of our red blood cell is permeable to urea. So what's going to happen when it's permeable? Well, urea is going to diffuse in, right? Because the concentration of urea in the red blood cell is 0 and it's higher in the bath. So it's going to diffuse in. For the red blood cell, there's going to be no change because all the ions inside the red blood cell are impermeable for the membrane. So what's going to happen? Well, our urea is going to diffuse in, right? And because it's, eventually inside our red blood cell, we're going to add on the 300 milliosmoles of the urea. So we get 600 milliosmoles. And the osmolarity of our urea bath stays the same, even though some has gone into the red blood cell. We're assuming that it's a really big urea bath, so it's not going to change much. So as you can see, we have a greater final osmolarity inside our red blood cell than the urea bath. And if we look at tonicity, right, we'll note that, so these are non-diffusibles. So because urea is a diffusible, we wouldn't count that in tonicity. So tonicity, we get 300 milliosmoles and 0 milliosmoles. So whether you look at final osmolarity or tonicity, you'll get the same result. So what's going to happen? Water is going to move into the red blood cell and you're going to get hemolysis and yes, brown P. Yeah, next slide. Yeah, so these are some questions, but because we're short on time, we'll skip through these so you guys can have a crack on your own. Yeah, so that's physiology for week 1.
Okay, so, wait, sorry. All right. Technically, this is HKS, but because we have gone through a very long lesson, I feel like, because this is a lot of reading more than anything, so I feel like I'll leave this for you guys to read. I'll just kind of go through some of it. So this is some of the WHO definitions. World Health Organization of health, equity, and health equity. Sorry for the images, I'll fix that before we upload it. But just have a read through it. The strength and limitations, that's also there. It's pretty straightforward if you read it. These are some of the things that last year's PSP had, but we did not get in our first year, our first lesson of HKS. But you do get this in Sem 2. So you could read through it if you don't have to, but these are just some of the terminologies that you'll be seeing more in Sem 2. Health inequities, geopolitics, environmental change, historical influential, social, cultural factors. All pretty straightforward, but these are basically all the things that you'll be talking about the things that affect global health systems and health inequities that occur within the health systems. This is an example of COVID-19 about how a health system should work. But basically the main point here that you'll be getting in HKS is that there are weak health systems out there. Our one's pretty strong. That's why we have 0 cases basically every day. But there are cases, there are a lot of countries which have very weak health systems. And these are where a lot of emerging diseases occur. And last year we got an example of Ebola. Coronavirus is also an example where you can see in countries which don't have the money and the time to put into a good healthcare system, you can see how the numbers for coronavirus is pretty high compared to places like us. We also got an example of rabies last year. And you guys probably heard of rabies, basically when the dog bites you, right? And the virus that rabies actually are from are called Lyssa virus. And it is very severe because once the symptoms are present, you can die. You basically die in 3 months. And there's also this really scary thing about hydrophobia, where because the virus is scared that you drinking water will kill them, they make the people who are the patient basically infected with the virus just be like scared of the fact of like having water. Like they will have like they'll shake themselves, like their vocal cords will be going weird and stuff. But yeah, it's a pretty scary thing, rabies. And the reason why this was an example last year is because this is part of a very big thing in India, where the vultures, which normally eat the cattle when they're dead, that's just normal, right? The vultures eat the dead people, or dead cattle. But the cattle were treated with these drugs that were actually toxic to the vultures. So the vultures would do their thing, eat the carrion, but then they would die because the thing that the cattle were treated with were toxic to them. That meant the carrion levels would rise. That would mean that instead of vultures eating them, the feral dogs would eat them. Then this would increase rabies. So you can see how in HKS, the main focus isn't just about how can you just treat this person with this medication. It's to see how there's a lot of different things affecting health in general, not even within the hospital, but just in daily lives as well, and in other politics, geography, social history, and all of that stuff. So in this case, we call this a trophic cascade because we basically removed one key species, and this has a really big effect on the ecosystem. Lastly, this is also a thing, rabies in the Sanjay Gandhi National Park in India. So in the city, there's this park where there's a lot of leopards. And these leopards, they basically kill dogs, that's their prey. But recently, because of the urbanization, the park has decreased, therefore the leopards can't actually just have their normal range, which meant that the dogs were free more, and that meant the levels of rabies would increase. So as you can see in this example and previously, HKS will tell you that medicine isn't just based on one factor. And there's a lot of different various components that are intertwined to create health outcomes. And that's basically HKS. It's a very interesting topic. We had a great lecturer last year. That was really interesting. But I'm not quite sure if you get the same person, but yeah. And that's it. Sorry for a very long lecture. Thank you for sticking by.
So thanks everyone. We're gonna try, and it's our first time doing it, so thanks for sticking with us. Next week we'll probably try to make it a bit shorter. We'll try to make it a bit more interactive. We'll talk amongst each other. But thanks for sticking through it. Come back next week. It was very exciting for us to give this to you, and we look forward to speaking to you throughout the year. So if you have any questions, you can stick around. Yes, hit us up on Facebook, DM us, whatever you need to do.
Thank you, guys.
Thank you very much. Thank you.
Thank you.
You guys.
Yeah, and just to reiterate, we'll be making a post with a link to our Google Drive, and all these slides will be up there as well. So yeah, don't stress.
And on a YouTube channel.
And YouTube video, yeah.
Yeah, no, thanks once again so much for you guys coming. Yeah, hope the rest of your week goes well.